you'd think that as organizers that we'd be perhaps the most exhausted people in this room, and to top it off, we're then asked to wrap everything up in a meeting that has been, I think, somewhat analogous to drinking from a fire hose. But before I go any further, I first of all want to really thank my co-organizer, Andrea Hevener. We started this journey, 18 months ago almost, to put together this meeting. And so we are relieved, we are exhilarated, and of course, very, very pleased with the outcome. I also want to thank all of the speakers who came and took part in the meeting, both from really all over the world, and that was wonderful. I also want to really make a point of thanking the students and trainees and those of you who submitted abstracts to this meeting. I would say that all talks were exciting, but some of the most exciting ones were the short talks from those abstracts that were submitted. So I want to really thank you and also acknowledge you for that as well. And then finally, to really thank all of you, the attendees, especially those of you who are staying here at 7:15 on the last day of the meeting, when there is food and nutrition happening. And it's very quiet next door, so it sounds like they again beat us. Nevertheless, we really thought that one of the things that we'd like to kind of share and convey in terms of a meeting wrap-up is really, first of all, to think back to sort of the early days of the mitochondria meetings. And I think one of the biggest changes over the last, 15 to 20 years in this field is that, the mitochondria is no longer really viewed as this static organelle that, makes ATP and makes some RAS and then, puffs out some cytochrome C to induce apoptosis. But I think that what this meeting has really taught us is that mitochondria are really very critical signaling hubs within the cells. So, we have extensive notes here in terms of what we learned from this meeting, and maybe I'll have Andrea start off and then I'll pick it up from there.
Well, I think, some of the important things that we learned about the signaling of the mitochondria is the release of mitokines and how they communicate within the cell as well as communicate to the periphery. And I think that's an exciting new area that we'll be expanding upon in the future and understanding, what is released by the mitochondria to signal to the nucleus or signal to other organelles. We also learned new approaches for mitochondrial therapeutics that take advantage of mitochondrial adaptations to disease states. We learned a lot about hypoxia, maybe as a therapeutic opportunity for certain mitochondrial disorders. We heard a lot about succinate and malonate and its involvement in ischemia reperfusion injury, and I thought that was really intriguing. The fact that certain metabolites can be used as signaling molecules and talk to the nucleus as well. Increasing ketone utilization in heart failure. One thing that I thought was really intriguing was the talk about gene editing to address inherited mitochondrial disorders. The ability to deliver zinc finger nucleases to the mitochondria was a really exciting talk for me because that's been a limitation in the field. And so I thought the application of that technology will be really interesting for future studies. Learning about other therapeutic strategies such as glutathione supplementation and knowledge in the regulation of mitochondrial dynamics, which is something that I'm intrigued by, how we control fission-fusion dynamics. And many of us have these mouse models where we're manipulating these regulatory proteins that control fission and fusion-mitophagy, and what that does to metabolic outcomes and phenotypes, I think is really intriguing. And we're really in the infancy in understanding these regulatory molecules. And I thought, in particular, Gerald Dorn's talk, where he very elegantly was able to manipulate certain amino acids to dissect apart the regulatory elements of the mitofusions, was a nice way to approach a research question scientifically and really start to delve into some of these clinically relevant questions that we're observing in human subjects.
Yes, I was also very impressed by the relationship between form and function. So we saw some really spectacular images of mitochondrial networks in skeletal muscle, in cardiac muscle, And really, the notion that the shape of the mitochondria not only reflects its response to outside cues or even its response to cues within the cell, but also might be correlated with the relationship between energy production in the mitochondria and where it needs to actually work in terms of contractile function or for example in terms of EC coupling. And I use that thought to segue into the fact that I think that the technology is really allowing us to image mitochondria both in animal models, and also there are some really interesting work in new imaging approaches that could even be used in living tissue. For example, the work looking at two-photon microscopy to actually look both in living animals in terms of things like NAD flux and so forth really represents, I think, an important advance. Of course, I think it's now really only scratching the surface. And one of the things that we're also sort of tasked with doing is, what do we see or where do we see the future going? And I think that, where we see the future going is more and more sophisticated methodologies and technologies to give us really much better imaging of mitochondria, both in vivo and ex vivo. And then in that regard, approaches to increase the resolution with which we can understand what actually happens to metabolites in mitochondria. You've heard quite a few presentations over the course of the meeting that really have used stable isotope labeling to essentially knock open very important new insights into where are the carbons going, and some of these studies have in fact challenged some of the dogma that perhaps we might have had when we had less refined tools. I'm looking at Zolt down there, and being the iconoclast that you are usually, and certainly the last thought that was presented, where you really used the ability to do very focused stable isotope tracing to really challenge perceptions in terms of mechanisms of action by which certain pathways might lead to metabolic modulation to have then an effect in terms of a pathophysiological state. So I think that moving forward we probably will see increased use of these approaches to really refine our understanding of many of these pathways and the crosstalk between small molecules and signal transduction pathway. Zolt is blushing down there, by the way, for those of you who can't see him face on. I think we also, in addition to that, really began to see how big data approaches were also being used to unravel certain of the mysteries about the relationship between mitochondria and aging and mitochondria and skeletal muscle function. I know that you're very keen on that approach as a future approach.
Very keen on that approach because I think you can use the large data sets to, especially when you perform metabolomics or RNA sequencing, you can mine those data sets and really look for novel regulatory nodes. And we've used that quite a lot. You heard a little bit about using the hybrid mouse diversity panel and looking at natural variation in gene expression. There was an elegant talk by Johan Arwix today. He uses a little bit different panel than we use, but we've been able to mine that to find gene relationships and gene trait relationships. And certainly we're going to be able to make those platforms available to the community to help you enhance your research as well. But big data science is really where we see a lot of the field heading, and then being able to collapse these data sets using more sophisticated statistical analyses like Bayesian, and we heard a little bit about machine learning, especially with the 3D imaging, which I thought was so elegant and really showing the heterogeneity of mitochondria within the muscle and being able to take those slices and collapse them and understand a little bit more about the mitochondria based upon where it's located, its morphology, and different functionalities of those morphological differences within the muscle, but certainly also comparative between muscle and other cell types, which Dale I think did a beautiful job in his OPA animals talking about how the mitochondria look different between cell types and some of the functionalities and the regulatory molecules that control the shape also lead to different metabolic outcomes and phenotypes. So I think that's an intriguing area.
So if we kind of put on our crystal ball now, so this is now, where is the field heading? So if we have this meeting again in 2 years, what will be the next sort of new hot thing that we'll all be flocking back to Keystone or wherever to hear about? So we're going to prognosticate, and this is, somewhat random, but, please feel free to challenge these prognostications as you hear about them. So I think we're going to see significant advances in just the tool, in our toolkits, that will enable us to dissect various aspects of mitochondrial biology. I certainly think that as well, we'll probably see applications of sort of more biophysical approaches to really drill down in terms of really how are mitochondrial membranes being deformed sort of in the kind of the mitochondrial dynamics part of our research. And, I think we'll actually see that in addition to the proteins that are involved in many of these processes, that there are probably also fundamental changes that will occur either in lipid composition that also tie into some of these changes. I think there's a lot more to be learned in terms of the signals by which mitochondria communicate both within the cell as well as across cells. And I think that we're really only beginning to scratch the surface in terms of what some of these signals are. And I suspect some of these are going to be additional small molecules, additional metabolic intermediates, peptides that are moving from the mitochondria to the nucleus and so on. And then I think that we're still at an early stage in terms of how do we target the mitochondria therapeutically. Quite a few people over the course of the meeting have really asked me, well, how feasible is it to drug the mitochondria? And my bias is I don't think it's particularly feasible, but I think what we can drug is pathways that are influenced by changes in mitochondrial biology or in mitochondrial metabolism. So I think that we'll be seeing significant advances in those areas as well over the next few years. So the final thing we're going to talk about before we send you out to have your final meal of this meeting is what are some of the opportunities and challenges. So it's nice to be very hopeful about where we might be a few years from now, but we also think that there are some real challenges that we'll probably have to overcome to even push the field forward. And I think that one challenge is as we again move into the area of how can we harness our knowledge of mitochondrial biology for therapeutic purposes. It raises the question that any systemic therapy potentially could hit mitochondria in multiple organs, which may not necessarily be what you want to do if the pathology is in one organ but not in another. So, one of the challenges which we are throwing out is how do you actually target therapeutic strategies in a tissue-specific manner? And so, we think that's potentially a challenge, but, something that we are maybe hopeful somewhat in this room or elsewhere may ultimately begin to crack, because I think that that would be a very important aspect of any kind of strategy that we really use to as a therapy, not necessarily for germline mitochondrial disorder, but for those very common disorders where the defect isn't necessarily in every single tissue or in every cell, but may in fact be focused on a given organ. I mean, I think that one exciting thing that Johan had in his talk was when he was manipulating the enzyme that regulates NAD. Really, the fact that there was significant difference in expression in, say, the kidney versus somewhere else, and that's by virtue of just the differences in expression pattern allowed a more generic approach to be somewhat more specific. But what I'm thinking really in terms of a bigger challenge is how do you actually take something that might be much more ubiquitous in terms of a pathway that clearly is implicated in one organ or tissue but not in another organ, how do you really target it where it ultimately needs to be targeted?
And then I think lastly for us, being in the School of Medicine, and I talked a lot about the translational relevance, making sure that our model organisms are, the mechanisms that we're describing in mouse models and lower organisms, our translation, that we can translate that knowledge to human subjects, because ultimately I think we all want to improve human health. And so at the end of the day, we need to work with our clinical colleagues and to really understand, what some of the conditions that they're trying to address in the clinic.
Yeah, and certainly I would, want to give a shout out to all of the speakers who actually showed us human data and human studies. The idea of having somebody volunteer to be biopsied, every 4 hours at night to look at mitochondrial dynamics. Well, I probably wouldn't sign up for that, but I guess in Holland, people will sign up for things much more than maybe in Iowa, where I come from. But I do think that having, really much more robust and rigorous studies in the relevant model organism, which is the human, for translating our findings to, improve human health and to obviously treat many clinical conditions, I think will be an important challenge to the field. And therefore, I think there's a lot that we all have to learn from each other in terms of how do we increase our collaborations with our clinical colleagues, and how do we also think about how we can, come up with tools that are hopefully, minimal invasive, or ways that we can really follow some of these very important pathways in our human subjects. So that's our sort of quick thinking and wrap-up of this meeting. And again, in closing, I really want to again thank all of you for very, very active participation in this meeting. The fact that you're staying here to listen to us drone on when, you could be out really, again, speaks either to the fact that you don't want to let me down, or maybe more importantly to the fact that, we'll all leave here exhausted, but rejuvenated in terms of the future of mitochondrial biology, and specifically as it relates to our favorite organs, which are varied, but I think this was a muscle and heart meeting, and I think that we are walking away with significant additional insights that will inform our future projects. So with that, I thank you very much, and wish you safe travels back to where you're going. And hopefully look forward to seeing you again at another similar meeting with new chairs and co-chairs. Thank you.
Thank you.